Hi my dear students, I'm Dr. Jeetu. Today we'll be dealing with our second chapter, Periodontal Microbiology. My references are Commensal's 13th edition and Linde's 6th edition. Linde's 6th edition have some extra points when compared to Commensal in this area. So it will be better if you can refer that also. I have included most of the important points. So going into the presentation. So we all know the oral cavity of a fetus is a sterile environment. Yes. When the baby comes out of the mother's womb, then microorganisms start colonizing the oral cavity of the newborn. So, which is the first organism to colonize the oral cavity is an important question. Streptococcus salivarius and Streptococcus mitis are identified as the first and dominant oral microorganisms. If you have salivarius as well as mitis in the options, go for Streptococcus salivarius. So, these are aerobic microorganisms. The second day, anaerobic microorganism can be detected in the infant's mouth. Then, after tooth eruption, a very complex oral microbiota will be established. Then, the species that colonizes the teeth will include Streptococcus sanguinis, Lactobacillus oralis, and all these species. Now, the microorganism of interest to a dentist, the S. mutans, which is a causative of dental caries. When is this colonizing the oral cavity? Again, an important question for you. Window of infectivity for accusation of S. mutans is 26 months. So, S. mutans window of infectivity is 26 months. So, the oral cavity will become a diverse environment for a number of bacteria. So naturally the plaque also will contain a variety of microorganisms, and how many microorganisms 1 gram of plaque will harbor? 1 gram of plaque will contain 10 raised to the power of 11 bacteria. So when a microorganism colonizes the oral cavity, it can colonize 6 niches inside the oral cavity. One is the intraoral supragingival hard surfaces. Supragingival it is. It can be teeth, it can be implant, it can be restoration processes, whatever it is supragingival. Second is the subgingival regions. So it can be the cemental surface, the implant surface. It has the presence of clavicular fluid and the pocket epithelium. After the supragingival and subgingival regions, third one is the buccal palatal epithelium. See the figure, the top portion, and the epithelium of the floor of the mouth, the bottom portion. The fourth niche is the dorsum of the tongue, 5 is the tonsil, and 6 is the saliva. So, supragingival surface, subgingival surface, buccal palatal epithelium and epithelium of the floor of the mouth. Then we have dorsum of the tongue, then tonsil, and finally saliva, which bathes all these niches. Coming to plaque or biofilm. We used to term this as plaque previously. Now, biofilm is considered a better term. What is biofilm? Biofilm is a structure in which bacterial cells are interspersed in a matrix. So, rather than considering bacteria as single units, it is considered as a community where different bacteria are living in a matrix. So this community lifestyle provides many benefits to the participating organisms. Since we are producing a intercellular matrix which is very diverse, the microorganisms habitat range becomes broad. A broader habitat range is there. Then there is food chain. That means the metabolic byproduct of some bacteria will be used as food by some other bacteria. So this establishes a food chain. And also there is gene transfer. Antibiotic resistance gene or better survival genes are transferred from one microorganism to another. Now we already said bacterial cells are interspersed in a intercellular matrix. So when you speak about the intercellular matrix, you can tell all the organic constituents that you know, the carbohydrates, proteins, lipids, DNA, albumin, glycoproteins. Okay. Among these, which are the important constituents? First important constituent is the glycoprotein, which is derived from saliva. And next one is polysaccharide from the bacteria. When speaking about the polysaccharide, there are two types, extracellular and intracellular. Which are the extracellular polysaccharides? Again, very important, the dextrans and the levans. They are very important in the structure of intercellular matrix. What are dextrans and levans? They are the glucose and the fructose moiety of the sucrose. We also have intracellular polysaccharides like amylopectin in the matrix structure. Coming to formation of plaque. This figure, you are going to get figure-based questions. So, be very familiar about the pictures. See the pictures repeatedly and also see what is written in every picture. Okay, they can hide some of these and they can ask what is the step. So, this is from Linde. it is a very simple figure actually, that you can see enamel, right? The rectangular purple colored boxes enamel. So the first step, it is numbered 1 and it is put in an oval box, is pellicle formation. So the salivary glycoproteins attaches to the enamel surface and forms the pellicle. Second step, you can see 2, 1. Okay, in your upper left corner you can see transport of the bacteria or passive transport of the bacteria towards the pellicle surface. And in 2, 2, you can see reversible attachment of the bacteria onto this pellicle layer. The second step, the bacteria gets transported to the tooth surface and it attaches reversibly onto the pellicle. Look for the third step. The third step is a irreversible attachment. Then you have co-adhesion. Co-adhesion means by this reversible and irreversible attachment, some bacteria will get attached to the tooth surface, right? So these bacteria will help some other bacteria colonize the oral cavity. This is called co-adhesion. And where is the fifth step? Fifth step can be seen on the bottom left corner. It is the biofilm maturation. Biofilm maturation means all these bacteria, because of the primary colonization, secondary colonization, everything, and multiplication of these cells, the biofilm matrix become more mature. And finally, the last step is detachment. After a limit, this will detach from the surface and it colonizes on some other tooth surface. Clear? We'll go into detail. So we'll brush that up. First step is adsorption of a conditioning film or pellicle. Second step is reversible adhesion between the microbe and the pellicle. Third step is permanent adhesion. How this permanent adhesion is happening? It is by specific molecules on the microbial surface called adhesins, and there are complementary molecules in the pellicle. So the irreversible attachment or the permanent attachment is using the adhesins of the bacteria. Now the fourth step, co-adhesion. As we said, the bacteria which attaches to the tooth surface first are the primary colonizers. The primary colonizers are usually aerobes. So they use up the oxygen and they make the environment anaerobic, so that secondary colonizers, which are usually anaerobes, can come and attach to this surface and they can live there. So this is co-adhesion, where the secondary colonizers attach to the receptors present on the already attached bacteria. The fifth step is plaque maturation. This is because of the multiplication of the attached cells and the increase in this biomass. And also there is synthesis of the matrix. That is how the plaque matures. And finally, as we said, there is detachment of these attached cells to promote colonization elsewhere. We will see each step. First, pellicle formation. So our pellicle is composed of 2 layers. There is a thin basal layer. This is very difficult to remove, even with chemical and mechanical treatment. Also, we have a thick globular layer attached on the top of the basal layer. Second step is initial adhesion or reversible attachment. Reversible attachment is using which forces? Again, important question for you. The reversible attachment is by the use of van der Waals forces, hydrogen forces, and electrostatic interactions. And who colonizes? The primary colonizers attaches by this reversible mechanism. It can be Haemophilus, Streptococci, Neisseria, Actinomyces, Veillonella, all these gram-positive aerobic organisms usually, and they will open up new binding sites for the adhesion of other oral bacteria. You should also remember a term called DLVO or Derjaguin, Landau, Verwey, Overbeek theory. This theory explains the behavior of the bacterial cells by the use of colloidal stability. Okay, so it comes under the reversible attachment of bacteria to the pellicle surface. Coming to irreversible attachment, we already mentioned it is by the use of adhesins. So one of the best characterized interaction is between which adhesin? Antigen 1 or 2 family adhesins of Streptococci and GP340 or glycoprotein 340, which is present in the pellicle. Antigen 1 or 2 family adhesins are usually shown by Streptococci. And GP340, from where it is coming? It is coming from the saliva naturally. It is a component of pellicle and it is derived from saliva. Fourth step was co-adhesion. The primary colonizing bacteria will provide new receptors for the attachment of other bacteria or secondary colonizers. This is known as co-adhesion. Now you have to differentiate between co-adhesion and co-aggregation. Co-adhesion, we already said primary colonizers comes and attaches on tooth surface. Then they will express some receptors to help the attachment of the secondary colonizers. But what is co-aggregation? It is like a direct interaction, or it is a mutual understanding. Here the cells are stuck together by some molecules in the solution. Many cells will come and club together, or they aggregate together. So at least 80 genera from the oral cavity shows some form of co-aggregation. When you talk about co-aggregation, the first bacteria you should remember is Fusobacterium. Why is Fusobacterium important? It can co-aggregate with all the other human oral bacteria. So whenever you see a clump or an aggregation of bacteria, you should suspect a Fusobacterium inside that mass, because it can co-aggregate with all the other bacteria. So many co-aggregations are helped by lectin-like adhesins, and they can be inhibited by lactose and other galactosides, or also by amino acids like arginine. So co-aggregation is helped by lectin-like adhesins and inhibited by lactose and galactosides and arginines. So we already said primary colonizers are Streptococcus, Neisseria, Veillonella, such species. Secondary colonizers are the proper periodontal pathogens that we usually study, like Prevotella intermedia, Prevotella losche, Capnocytophaga, Fusobacterium nucleatum, P. gingivalis. All these species, they cannot colonize a clean tooth surface, but they require the primary colonizers to adhere to the tooth surface, and then they help in the attachment of these bacteria. So, when more than 13,000 plaque samples were analyzed, 40 subgingival microorganisms were found, okay, and they were classified under color-coded complexes. We already know there are many color-coded complexes: red complex, green complex, yellow complex, orange complex. So, this different complexes names was based on the frequency with which the clusters of microorganisms were recovered. We do DNA hybridization at certain frequencies. A group of microorganisms were recovered. So these organisms were clubbed inside that particular color complex. Clear? We have blue complex, which includes actinomyces species. Then we have yellow complex. Yellow complex composed of Streptococcus species. Streptococcus mitis, sanguis, oralis, basically. Then we have purple complex. Purple complex have two bacteria, V. parula and A. odontolyticus, actinomyces odontolyticus. This yellow complex and purple complex are found in health. So, periodontal health composes of yellow complex microorganisms and purple complex microorganisms. Next, we have green complex. Green complex can be remembered using the acronym ACE. ACE, one is AA, or Actinobacillus actinomycetem commitans. Okay, the term has been changed to Aggregatibacter actinomycetem commitans recently. So this is the new term for AA. After that, C we have Capnocytophaga species, and E stands for Eikenella corrodens. So this is green complex ACE, AA, Capnocytophaga and Eikenella corrodens. Then we have orange complex. Orange complex is comprised of Fusobacterium nucleatum, then Campylobacter rectus and Prevotella intermedia. Then finally we have the red complex. Red complex has Porphyromonas gingivalis or P. gingivalis, Tannerella forsythia and Treponema dent. We've brushed that up. Blue complex, actinomyces species. Yellow complex, composed of Streptococcus species, basically mitis, sanguis and oralis species. Purple complex, V. parvula and A. odontolyticus. Red complex can be remembered using the acronym PTT. We know red reminds us of blood, and PTT is a test for blood clotting, right? So, P. gingivalis, T. denticola and Tannerella forsythia, PTT. Then orange complex can be remembered using the acronym NRI, nucleatum, rectus and intermedia, NRI. And green complex ACE, we already said AA or Aggregatibacter actinomycetem commitans, Capnocytophaga and Eikenella corrodens. That's all about the color complexes. So we said there are numerous bacteria. There is 10 raised to 11 bacteria in 1 gram of plaque, and they exist in different color-coded complexes. We also have non-bacterial organisms in the plaque. We have viruses. The group of herpes viruses, which are enveloped double-stranded DNA viruses. They have 8 different members. So there are 8 different species of herpes virus. We have 5 genera of human papillomavirus. Papillomavirus are also double-stranded DNA viruses, and they are non-enveloped. Herpes virus was enveloped. Papillomavirus is non-enveloped. Okay. Then we have picornaviruses. Picornavirus is also a non-enveloped virus, but it is single-stranded RNA virus. Then we have retrovirus, which is single-stranded RNA virus again, but enveloped. There are 7 genera of the retroviruses. Then we have fungi. We have Candida albicans, which is the most common opportunistic fungal organism. Not just Candida albicans, we also have Candida tropicalis, glabrata, cruzae, dubliniensis, and most of the Candida species are there. We also have saprophytes, which cause local infections like Entamoeba gingivalis and Trichomonas tenax. Then it doesn't end with viruses, fungus, and saprophytes. We have parasites also. We have systemic parasitic infection causing organisms like Leishmania. And finally, archaea species. Archaea are single-celled organisms, but they are not considered as prokaryotes. They are considered as eukaryotes. So can you believe our plaque consists of these many organisms? So do you remember in which step of plaque formation we were in? We finished pellicle formation, reversible attachment, irreversible attachment by the bacteria, and we learned other than bacteria there are so many other microorganisms in the plaque. After this, we have to go and see about plaque maturation. And we already said during plaque maturation there is a development of food chain, wherein end product of metabolism of one organism is used as the nutrient by another organism. We also have cell-to-cell signaling. Plaque bacteria communicates with each other in a cell density dependent manner. Cell density dependent manner means when there are more number of the cellular communication molecules, there is a stronger communication. So in Streptococcus mutans, this communication is known as quorum sensing. So quorum sensing is mediated by CSP, or competence stimulating peptide. Usually it is released by gram-positive organisms during their growth. And there is another signaling molecule. It is considered as a universal signaling molecule, which is known as autoinducer 2. So there are 2 main signaling molecules. One is CSP. It is mainly secreted by Streptococcus, and one more is autoinducer 2. It is secreted by many other microorganisms, and it is considered as a universal signaling molecule. And which gene codes for this universal signaling molecule autoinducer 2? Lux S gene codes for this AI2 molecule. So this is a picture of food chain. You don't have to by heart where all these arrow marks are going, but the main things you will have to remember. So I'll tell you the main things. I have encircled the major things. Okay, the Streptococcus or the actinomyces, which are the primary colonizers, they produce lactate, which is a product of their metabolism. So I think all of you know a brand called Lavi, right? For bags we have a brand known as Lavi. So I remember it this way, Lavi. Lactate is taken up by Veillonella. So our Streptococcus and our actinomyces is producing lactate, which is utilized by Veillonella. These organisms also produce formate, which is utilized by the Campylobacter. Formally going for a camp. Formate is taken up by Campylobacter. So with our Lavi bag, we are going formally for a camp. So lactate produced by the Streptococcus or actinomyces is taken up by Veillonella. Formate produced by Streptococcus or actinomyces is taken up by Campylobacter. I forgot to tell you something. The highlight of this thing is I have put it inside the red box, or PG, or our P. gingivalis, which is a keystone pathogen, snatches nutrients from everyone. So finally, everything you can notice, the arrow marks, everything is coming towards the PG. Rarely it gives off some metabolic byproduct to someone else. So this Veillonella species, our Lavi and formally going for the camp, you should remember, lactate taken up by Veillonella. Veillonella produces menadione and gives to PG. Again, our formate was taken up by Campylobacter, and this Campylobacter prepares protoheme and gives to PG again. So this much is clear for you. And on the right side, I have encircled something. Capnocytophaga species prepares succinate and gives to PG. How will you remember this? You pull up your cap when you get success, right? So Capnocytophaga produces succinate, which is used by PG. Then one more interaction that you should learn, I have put it on the right side. Read it as PITS. You can see it, right? PITS. That is, P. gingivalis produces isobutyrate, which is taken up by Treponema species. Treponema species produces succinate, which is again taken up by Porphyromonas gingivalis. So this cycle, you should remember it as PITS. I hope this three major interactions are clear. So we were talking about plaque maturation. There are some structures or cell associations which we can see in a mature dental plaque. One is corn cob formation and another one is test tube brush formation. Corn cob, we all know what is a corn cob, right? Corn cob structure, there is a central long portion and surrounding this we have round corn kernels, right? Just like that, there is a filamentous organism, and around this filamentous organism we have a number of coccal cells attaching on this. You can see the figure. Classically, the Streptococcus species, which are cocci, right, which attach to the filamentous organism, Corynebacterium matricioti or actinomycetem species. The corn cob formation is important. Another structure is test tube brush. We all have seen what is a test tube brush. So, in a test tube brush, the central organism is a filamentous organism, and there are rod-shaped bacteria sticking out perpendicularly from this filamentous organism. So, characteristic association in a mature plaque is corn cob formation as well as test tube brush formation. Now we saw how plaque is formed, right? Now there are some things about the plaque formation, the rate of it, where it is formed more, when it is formed more. Okay, we'll go to that. The rate of de novo plaque formation is similar in young and old patients. When we are examining the oral cavity of patients, often we would have noticed older patients have more plaque in their oral cavity. So we tend to think that older patients have a more rate of plaque formation, which is wrong. Rate of plaque formation is the same in young as well as old patients. Then why do we see more plaque in the older patients? Because their manual dexterity to clean this plaque is lesser, right? So that is why they retain more plaque, but the formation is in the same rate, in the same pace. Okay, now plaque formation is more in the lower jaw as compared with the upper jaw. It is more in the mandible. Okay, in the mandible only, where it is more in the molar area when compared to the anteriors. Again, in the molar area, where on the buccal surface as compared with the palatal surface. And finally, it is more in the interdental region as compared with the buccal or the lingual surface. So more in the mandible, more in the molar, more in the buccal than in the palatal, more in the interdental than in the buccal or palatal. One more thing is, how is the plaque growth during day and night? What is the difference? During night, the plaque growth rate is reduced by 50%. Why plaque is less during night? Because during night, salivary production is less, and principally our plaque formation requires salivary proteins for pellicle formation. That was our first step, right? Pellicle formation. So when saliva decreases, naturally plaque formation rate also decreases. Now I want to bring to your notice two confusing terms: allogenic succession and autogenic succession. What is allogenic succession? A change in the composition of a bacterial community due to a non-bacterial or non-microbial factor is called allogenic succession. We know that auto means same and allo means different. So allo means it is not due to a bacterial factor, it is due to a different factor. For example, smoking. Smoking is a non-microbial factor which can alter the composition of the bacterial community. So this is an example of allogenic succession. Autogenic succession means auto or same or self. So change in the composition of a microbial community due to microbial activity itself. This is autogenic succession. One more important question. So whatever is happening inside the plaque, whether it is autogenic succession, allogenic succession, the plaque becomes clinically visible after 36 hours. like 1.5 days, right? So plaque becomes clinically visible in 36 hours. In green complex, we talked about AA, right? Aggregatibacter, actinomycetem, commitans. AA is attributed as a major causative organism for aggressive periodontitis. So, questions about AA which can be asked are, what type of colonies AA forms? AA forms star-shaped colonies when they are embedded in agar. And one more thing is periodate. What is periodate? Periodate disrupts the carbohydrate structures. So periodate disrupts AA from the tooth surface. And one more thing about AA is translocation. Translocation means when we probe a particular tooth and AA was present on that tooth, this will get attached to that probe surface. And when you probe another tooth surface, this AA colonizes the newer tooth surface which was uninfected. So this translocation of AA was demonstrated by Christensen. So these are the three important questions that can be asked about AA. Coming to plaque hypothesis, we have non-specific plaque hypothesis which was given first by Miller and it was modified by Thiele. Non-specific means we are not concerned about the specificity of the plaque. Whatever is the composition of the plaque, if plaque is present, disease will be present. This was what Miller said. Then in an updated non-specific plaque hypothesis, Thiele was proposing when plaque is present, it causes disease definitely. But one thing is that severity varies. That means some plaque causes gingivitis and some plaque can cause periodontitis. So this is the non-specific plaque hypothesis. Any plaque is pathogenic. That is the essence. The second plaque hypothesis is specific plaque hypothesis. This was accepted because of the recognition of AA as the causative organism in aggressive periodontitis. So they gave importance to the specificity of the plaque. If the plaque is composed of relatively non-pathogenic organisms, it will not cause disease, and specific microorganisms when present causes specific disease. So that is what is specific plaque hypothesis. All plaque are not pathogenic and it depends upon the composition of the plaque. It was given by Losch. The third plaque hypothesis is ecologic plaque hypothesis. He tried to unify the existing theories and he said it is not just the plaque, it also depends upon the environment, whether disease will be caused or not. It was given by Marsh. Then we have keystone pathogen hypothesis. It was given by George Hagi Shenkalis and he attributed a keystone role to our PG or P. gingivalis was considered as the keystone microorganism which causes the disease. And finally, we have the PSD or the polymicrobial synergy and dysbiosis hypothesis. This theory also considers PG as a keystone pathogen, but proposes that PG causes dysbiosis in a polymicrobial community which was under synergy. The polymicrobial or the multiple microbial community was in a synergy and PG disrupts this synergy and causes dysbiosis in the community. It does this by three things. TLR subversion. TLR or toll-like receptor for subversion. TLR is a component which is important for our innate immunity. So this TLR subversion happens inside the host. That means PG acts as an agonist of the TLR4. So this will activate the immune system. And sometimes PG acts as the antagonist of TLR4. This will dampen the immune system. This depends upon the concentration of iron. So it is antagonizing TLR4 and it is decreasing our immune response. We can understand it, right? Then why is it acting as an agonist and activating the immune system? If it is doing so, will not the immune system kill the PG? Yeah, you got that doubt? If you got that doubt, it's a genuine doubt. But the thing is, the PG act as an agonist for the toll-like receptor. It activates the immune system, but it makes sure that the immune system will not kill the PG. So it causes TLR subversion is the first mechanism. Second thing is local chemokine paralysis. There is a paralysis of the chemokine interleukin 8 or there is inhibition of synthesis of interleukin 8. And third one is activation of C5A. So just like the TLR4 activation, it activates C5A, but it will make sure that this will not kill the PG. So this is how the polymicrobial synergy and dysbiosis is happening by TLR subversion, local chemokine paralysis, and activation of the complement factor C5. Coming to the disease progression models, we have three disease progression models: continuous model, random burst model, and asynchronous multiple burst models. You have to be thorough about the figures also, which figure denotes which model. The first one is the continuous model. This model states that wherever plaque is present, there is destruction or disease happening in a continuous and a time dependent manner. You can see from the figure, as time increases, the destruction is also increasing in a continuous manner. So this is the continuous model. The second one is random burst model. Usually, random burst and asynchronous multiple burst is little confusing. You should remember it like this. In random burst, everything is random. Which site will get disease is random. The extent of destruction is random. Rate of destruction is random. Everything is random. In asynchronous multiple burst model, there is a burst of disease activity for a finite amount of time. Then there is a period of inactivity. There is a prolonged period of inactivity. And this also, some sites will get affected and sometimes will not get affected with disease during any time. So be clear with the figures. So once again, random burst model, activity occurs at random in any site. Some sites show no activity, some sites show one or several bursts of activity, and the extent of destruction also varies among sites. Asynchronous multiple burst model, several sites show burst of activity over a finite period of time and followed by a prolonged period of inactivity. Occasional bursts occur infrequently at certain sites. Other sites show no periodontal disease activity at any time. What is the difference between random burst and asynchronous multiple burst? Okay, in asynchronous multiple burst, the majority of the destructive activity take place within a few years of the individual's life. We already said, okay, there is a burst of activity during a particular finite period of time. Then there is a prolonged period of inactivity, something like the aggressive periodontitis scenario. There is a burst of activity during a finite period of time. Then there will be a prolonged time of inactivity. Now we have some postulates to decide whether an organism is a pathogen, right? These are known as Coates postulates. We have four postulates. One is it should be isolated from the deceased individual. If we are getting that from a deceased individual, then only we can say this is pathogenic. Two, after getting this from the deceased individual, it should be grown in a pure culture in lab. We should be able to grow it in the lab. Third one, when it is taken and inoculated into another organism, it should develop disease. And fourth one, we should be able to recover this from a lesion of this lab animal. So one thing is we should get it from a deceased individual. Second thing is we should be able to culture it in a lab. Third one, when taken from the lab and injected in another organism, that organism should develop disease and we should be able to recover this organism from a lesion of this lab animal. In periodontics, this is modified as Sokransky's criteria. So Sokransky has given 5 criteria, that is association, elimination, host response, animal models, and virulence factors. Something similar only it is association. The organism should be associated with that disease. 2. Elimination. When the organism is eliminated, the disease also should get eliminated. Third one is host response. When this organism is present, the host should elicit some response against this organism. Fourth one is animal models. Again, it should be demonstrable in animal models. And 5. Virulence factors. These potential periodontal pathogens should be able to express virulence factors. What are virulence factors? There are 3 types of virulence factors. One is factors that can promote the colonization of the bacteria. Whichever things which will help the bacteria to colonize are virulence factors. Examples are adhesins. After they colonize, what they'll do, they'll produce toxins and enzymes to degrade the host tissues. So that is the second virulence factor, toxins and enzymes to destruct the host tissue. And third one is whenever the microorganism is colonizing the host will produce immune response against the bacteria. So the mechanisms that protect this bacteria from the host is the third virulence factor of the bacteria. Okay. We learned the major virulence factors of major periodontal pathogens. Treponema denticola, which is part of the red complex, has its major virulence factor as major sheath protein. MSP or major sheath protein is a virulence factor of T. denticola. Now, trypsin-like proteases. This virulence factor is produced by P. gingivalis, T. denticola and Tannerella forsythia, or all the red complex bacteria produces trypsin-like proteases. Our P. gingivalis specifically produce enzymes known as gingipains. There are three types of gingipains. We have R gingipains and K gingipains. R gingipains are arginine gingipains. They are of two types, R gingipain A and R gingipain B, and K gingipains are lysine gingipains. It is depending on their ability to cleave the bond, arginine bond or lysine bond. If it cleaves the arginine bond, it is R gingipain. If it lyses the lysine bond, it is K gingipain. Okay. Then we have a virulence factor known as leukotoxin, which is produced by our AA. Why it is known as leukotoxin? Because it is toxic to the human leukocytes. What will it do? It will catalyze the conversion of all the ATP of our leukocyte to cyclic AMP. It converts all the ATP of our leukocytes to cyclic AMP. Hence, it is leukotoxic. When an AA produces so much of leukotoxin, which is known as a hyperleukotoxic strain. An example of a hyperleukotoxic strain is JP2 or JP2 clone of the AA, and it is uniquely associated with localized aggressive periodontitis. So JP2 clone is a hyperleukotoxic strain, and it is specifically found in localized aggressive periodontitis. I will tell you one more new term and we'll wind this up. It is known as Commensal paradox. In spite of a host having array of or a big sequence of immune inflammatory responses, many microorganisms are coexisting in the body without initiating any such response. This is known as Commensal paradox. So it looks like a paradox, right? In spite of having so much of immune inflammatory response, some microorganisms just staying in its own house, and we are not bothered also. This paradox is known as the Commensal paradox. Why this is happening? That is because the resident oral microbiota, you can see the center box, right? They help us in many ways. So that is given in the four outer boxes. One is nitrate NO pathway. That is, the gastrointestinal and cardiovascular benefits due to the NO is produced to the host by these microorganisms. The nitrate NO conversion is brought about by the microorganisms, and this helps us in modulating the blood pressure, the blood flow, to stimulation of the gastric mucus and for the inhibition of other pathogens. So one thing is nitrate NO conversion. Second thing is colonization resistance. Clear from the term. Because of the colonization of these resident microbiota, it is resisting the colonization of other pathogenic microorganisms, so that it will compete for nutrients, compete for adhesion sites, and secretion of the antagonist against the pathogens, and creates unfavorable growth conditions for the pathogens. Third one is host development. Many commensals or many resident oral microorganisms contributes to the development of the host by stimulating its host defenses, contributing to digestion, etc. And finally, we have a host crosstalk. That is, these commensals or the resident microbiota crosstalks to the host and tells it that don't go for pro-inflammatory responses. I am not your enemy. I am not going to harm you. And it downregulates the pro-inflammatory response, and it stimulates the interferon responses, or it is a more protective responses. So this is the Commensal paradox. That's all. You may feel it a little too much. You may feel it is more than what is needed, but nowadays the question papers are looking like PG level question papers. They want finer filters, so they ask more detailed questions. Don't worry, I have explained it in simple terms. Just understand what is what, okay, and repeatedly see the figures, and repeatedly you bring to your mind the key points. Get a key point about something and remember that, so that you understand the entire thing and you don't get confused. Okay, if you have any doubt, you can contact me. Thank you.